So if it were AUG, what would you have? You look in the table, it will be met. Okay, so what about CGC? CGC, arginine, CGC, okay, UCA, serine, CGU, CGU is glycine, GAU, aspartic acid, ASP. What about UGA? Oh, sorry, CUA. So which one did I read? This one is correct.
The next one. Yeah.
Okay, GCA is what? GCA.
Yeah.
GCA is what? Alanine. And CUA. Leucine. ACU. Look under A. ACU is 309. THRT. UGA, you go away, nothing, right? So it's only how many amino acids long? 6 amino acid long protein. Okay. So the other page, basically they have given you the reverse, they have given you the amino acids. So if you want, you can fill in the anticodons. They have given the mRNA codons and the DNA template also. So this one you have to go backward. The other one you did from the top, right? From DNA to mRNA to tRNA to the amino acid. So this page, this example is the other way around. And then there are some questions like what carries the genetic code to the ribosomes?
RNA?
What RNA?
mRNA, yeah.
This is formed of an mRNA-based triplet. What is that? Codon.
Codon.
Specifies a particular amino acid. Codon. Sites of protein synthesis. Ribosome and ER. Rough ER. Carries amino acid to the ribosome. What carries the amino acid to the ribosome? tRNA. It transfers the amino acid to the ribosome. tRNA triplet that pairs with the codon.
What would you at?
I am at number 6.
Number 6.
You got
Anticodon. Codon that starts polypeptide synthesis. We know it's the start codon, but which one is that?
AUG.
Codon that stops, there are 3 possibilities. UGA, UAG, UAA. Then they have asked you they have given the amino acid and then they have asked for the DNA based triplet and anticodon. So how can you find that if you were to do it? How will you proceed? You have the amino acid, you have asked for the DNA triplet and the anticodon. So how will you proceed? What do you need to know first? You have the amino acid information. Okay, where do you get it from?
Table.
Page Page 2. But basically from the table, you know, those three nucleotides code for the amino acids. What do you call them, those three nucleotides? Where did this come from, or where is this coming from, this table? mRNA codons. So that's what you need to do first. And then if you know that, you know the others. Because if you know your mRNA codons for these amino acids, and the complementary triplet on the DNA will be this column, and the whatever is complementary to the mRNA will be the next column, the TR anticodon, right? There are two columns that are blank, right? And the very last question, they have asked the possible sequences of mRNA codons that code for the polypeptide. So they have given the amino acids, and you need to find the codons from the table. So this exercise you can complete on your own. That will help you to understand this part better. Okay.
Can you do one example, like with the tryptophan? Just like
Tryptophan, okay.
UGG.
You already they have given you the mRNA codon. Even if they have not given, all you have to do is go up and look up the tryptophan on the table. Where do we have tryptophan?
It's UGG.
UGG. There's only one. So that's what they have already given you. So your
What do you do for ones that have more than one? You just pick one?
yeah, you can have both options.
Okay.
So you will have two sets of data.
Okay.
So what would be the anticodon be for the first one?
Which one?
How do you pronounce that?
Tryptophan.
Tryptophan.
This is a tRNA, right? So you have to use the AUCG rule. So you will have ACC.
It would be ACC. All right. Oh, okay.
And going backward from the mRNA, what would be the DNA template? You have U, the first place in the mRNA, so what would be on the DNA? A. Okay. Remember, in place of T only you have U, right? So it cannot be T against U. It has to be A on the DNA. So it will be A, C, C.
So the base triplet and the anticodon are the same?
In this case, it happens to be the same.
Okay.
In this case, because of the way it is. If you want, you can try one more. So let's look at valine, GUU. Okay. So the anticodon will be what? CAA. And your DNA template, you will have CAA. Okay, let's do one more. Methionine. You have AUG, right? What will be the anticodon? It will be UAC. Okay? Right? Now let's look at the DNA. TAC, they are not the same. Okay, quickly, what are the major differences between the DNA and RNA in terms of first the number of strands?
DNA is larger.
Number of strands, 2 versus 1. Okay, and what about the sugar between the two? What's the difference? What sugar you find in DNA and RNA? You know you have a sugar phosphate, yeah. You have a sugar phosphate on the base, right? So the sugar is different, ribose and deoxyribose, depending on whether it's DNA, RNA. That's how they get the name. And other major difference is the nucleotide. You have uracil in place of thymine. Okay. So replication is the copying of the DNA, one strand at a time. One of the strands is copied continuously, other strand is copied in pieces and then they are combined together. What is transcription? Translation. Transcription is the formation of mRNA using DNA as the template, as you did. And translation is the formation of your polypeptide based on your mRNA codons. Okay, in association with the tRNA anticodons. Okay, now let's go to your book and we will continue from there. Okay, let's look at page 134 that talks about karyotype. Karyotype is a display of your chromosomes or the chromosomes of any organism. So here you can see 23 pairs. As you can see in that figure 417, a normal human male. The male-female is determined by the pair number 23, which we call sex chromosomes. If they are XX, it's female, XY, it's male. And you can notice the X is larger than the Y as such. And then you have autosomes. They are pairs 1 through 22, they are called autosomes. And the pair 23 is the sex chromosome. Homologous chromosomes means the chromosomes are identical. Then you see germ cells and somatic cells. What are germ cells? Germ cells are the cells that give rise to the gametes, your sexual gametes or sex cells, namely the sperm and the ovum. All the rest they call somatic cells, the cells that make your bones, muscles, and other tissue. Then you have genes and alleles. So if you have a long stretch of DNA, it's part of your chromosome, okay? Your genes are located here and there. Unlike in bacteria, they are more or less continuous or overlapping. Here they are, like in one picture you had the A, B, C, D, E in pink, right? You have seen that figure. So these are all your genes. The location of the gene is the locus. And we call the copy of the gene, see, one came from male, one came from female. That's why you have a pair all the time, right? So the copies, the mirror images of the particular gene, we call them alleles. So one allele from mom, one from dad. Then dominant and recessive. Sometimes depending on the trait or the characteristic that the gene is coding for, it could be dominant or recessive. This is by nature. Okay. So how do you understand dominant and recessive? Okay. If I have a green sheet of paper like the what do you call the something that you wrap sweets and all, something light can pass through, or even any colored paper, craft paper or something. I wish I had two. Okay. So if you put green and white, look under the light, what color you will see?
Green.
Green. Because green is dominant. Put green and green, green. But put white and white,
Green.
white and white, green. You'll just see white. If you put two white sheets and look against the light, you'll see only white. Why? The green is gone now. Okay? And you have a tall guy, let's say Andrew, who's shorter than him. I don't know, maybe myself. I'm standing behind. As long as this guy is in front of me, you don't see me. Right? Yeah, so if you kick him out, then you'll see me. Alright? So that's an example for dominant allele. So sometimes, as long as this gene product is present, you don't see the expression of this. So you can try to relate that to understand what is a dominant and recessive allele. And we earlier saw the word homologous. Now you see homozygous in the next paragraph. further down on the page. Homozygous, heterozygous. Homologous means both chromosomes are identical at the level of the entire chromosome. Homozygous is at the level of the alleles. If they are identical, that's homozygous in that locus or location. If they are different, even a little bit, and even in one nucleotide, heterozygous. Then you have genotype, phenotype. Genotype is what you are made of, you have no control, but what you express is phenotype. So ideally, we all have genetic predisposition to be whatever we are, but depending on our circumstances and exposure to many things, we develop like skin color, everything. If you live in cold place year long, you'll be lighter versus some place where it's too hot, you may be darker, and that gets passed on also through generations and so on. And also the way your brain develops also, we are all born with the same Play-Doh. Your brain is like that. But how we make and shape depends on many factors. So genotype is the genetic makeup. The phenotype is what is expressed on the outside. What you see, it is observable. So a gene will be expressed, meaning only if it expresses the phenotype. So like this recessive allele, if you have a recessive allele, then sometimes both are dominant, sometimes one is dominant and one is recessive. So the recessive will only express if this gene is not there, meaning both are the same kind of recessive alleles. So for certain characters in nature, it has to be either dominant or recessive. Sometimes you have codominance, both of them are dominant, so you need both to express that. Maybe a color of some body part or something, so you need the product from this and that, they mix and match. And then they have some variations of the same thing, like multiple alleles, codominance, incomplete dominance. It was that I was just talking about. Sometimes you have more than two. There are other alleles from maybe some other location or locus that are responsible. So like in figure 419, you can see they call it polygenic inheritance. More than one gene is involved. Many genes are involved in eye color, for example. Then you have codominance, when both alleles are dominant equally, they call it codominance. Or sometimes when you have these two alleles coding for one product, but when you combine them, you get a totally new product, that is incomplete dominance.
You mean the two product combined?
Yeah.
Like both parents
Like
have
if,
brown hair, brown eyes,
right.
and then they've got kids, their kids
Yeah,
end up with
like-
blue eyes.
Yeah, so brown and blue, you get a different color when you mix.
Okay.
Polygenic inheritance, multiple genes are involved, polysaccharide, polypeptide, we talked about that. Pleiotropy is the opposite of polygenic, meaning polygenic, many genes are involved in one trait or one characteristic. Here you have one gene that can give different products or different characteristics or traits. So you can look at 420. There is 3 situations: darkened skin, darkened sclera of the eye, darkening of the urine. All this can be controlled by one gene as pleiotropy. It sounds like poly, this is pleo. It also tells you many what, but in the polygenic inheritance, you see many genes involved in one trait. Here, one gene creating 3 different effects. Sex-linked traits in the sex chromosomes, the pair number 23. Sometimes you have genes that are responsible for some traits. They call them sex-linked traits on the chromosomes. Already we discussed that they are sex chromosomes. So for example, color blindness is carried in the sex chromosomes. So you can see here, you see the female XX, you have the genotype is CC. On one you have the upper C, and the other X you have the lower C. So the 1C is dominant, right? So that takes care of the whatever you need for normal vision. But if you see in a male, if you are having only the lowercase c, you don't see the uppercase c, so something is missing. If that is the case, as long as you have an uppercase C, so that will dominate and the vision will be normal. But in case of some color blindness, you will see the recessive gene expressing itself. That should be good for that chapter. Okay. So we will do a little bit of histology. Yeah. Not the whole chapter. First of all, you need to know what is histology, study of tissues. What is a tissue? What is a tissue? Anybody?
Group of cells working together.
Group of cells working together to do specific functions.
And
Right?
common function, right?
Then you have, in any tissue if you take, you may find some, yeah.
Is anything we go over in chapter 5 going to be on the test?
Yes, that's why I'm doing it now. Yeah, if you see my schedule, it includes chapter 5, but not the whole chapter, but whatever we cover today.
I got a question real quick. I have my buddy, he was colorblind and so was his father. Is it just by chance that that happened then? Because if you can only inherit it from your mom?
I cannot tell you, I'm not a geneticist, so I cannot
Just-
give you.
just
I
how
know,
the
see,
book is.
some of these questions I can probably help you outside class because
Yeah.
I'm not a medical doctor and I don't want to give answers which I'm not comfortable. I don't mind answering you
All right.
with my best guess.
All
So
right.
I know you are always coming up with some clinical questions. Otherwise, I will be teaching clinical pathology, not anatomy. So
All right.
I'm not upsetting you. Okay?
Oh, no, I
Yeah,
understand.
sure. So if you don't mind,
That's
we'll
fine.
get into that later.
All right.
So any tissue if you take, there are certain structures that you need to be looking for. If you take a section of the tissue and look it under the microscope, okay, you will find a background that's going to be called your ground substance, as is highlighted, or matrix, okay. So in the matrix, you may find some cells with the nucleus or some fibers, or some tissue, you may just find only cells. In some tissue, you may find a few or more fibers and just a few cells compared to this, this, and this. Okay. And then you should know what is ICF, ECF by now, intracellular fluid, extracellular fluid, inside and outside the cell. Outside the cell, extracellular fluid is between cells, so they may also call it interstitial fluid. So it's the same as extracellular fluid. So if you take a cell, okay, there's a cell, there's another cell, there's another cell. ICF is your cytosol inside the cell. So whatever liquid that you find in between, that's your extracellular fluid, or also they call it interstitial fluid. Then embryonic tissues, these are the tissue layers first developing when you form a baby, before even it becomes a baby. So they call these layers germ layers. So if you have a sperm and ovum, they fuse, they form what they call a zygote, like a single cell. It becomes 2 cells, 4 cells, 8 cells, 16 cells, 32 cells. What kind of cell division we are talking about when you do this? Go from 2 to 4 to 8 to 16. What kind of cell division? Mitosis. Exactly. Okay. So when you come here up to this point, then it starts differentiating instead of just dividing and growing into different layers. They call them germ layers. The outermost layer will be the ectoderm. You see those words, ecto, endo, and mesoderm. Ectoderm is this outer layer, more inside, endo, in the middle, in between is the mesoderm. These are the first 3 germ layers. Later, the ectoderm will become your epidermis, the skin. Okay. The endoderm will become your muscles, bones, and blood. Mesoderm will become the inner linings of the organs, some of the internal organs. But all I want you to understand is what is the primary germ layer, what are the 3 different regions. Then you have to prepare the tissue in order to see how they look like under the microscope. So they section, meaning cut it across, and you can cut it in different ways. They call these histological sections. So if you see figure 5.1, what do you see in the 3 different figures? If you don't cut it right, you'll be missing something. Look at the egg or look at the tube, right? So if you have to cut it right, it has to be right where you can see all the parts. Okay. And then there are names for the different sections in page 146, figure 5.2. On the top, you see a bone that is cut right across from the top. They call it LS, longitudinal section. Lengthwise, L for lengthwise, L for longitudinal. The other one, in the transverse orientation, if you cut, they call it transverse section or cross section. You're cutting across, right? And then the third one, you're cutting diagonally. That's called oblique section. So you can have longitudinal, cross, or transverse, or oblique sections. You can have smears and spreads. Smear is if you take a liquid sample and spread it, or you can take a small section of the tissue and you can spread it on a slide. Then you have the epithelial tissue. You have 4 major categories of tissue. First is your epithelial. Then you have connective. Cartilage will be part of the connective tissue, and also blood and bone. Blood is liquid, bone is solid, unlike the other tissues which have a more soft composition. Then you have muscular tissue. And then the last one is the nervous tissue that includes your brain, spinal cord, and the nerves. Spinal cord and the nerves. So you have 4 major categories of tissues. But connective tissue, within that you have another one which we call cartilage that joins your bones in the joints. It's like ropes tying the bones together. Blood and bone are also considered as connective tissue. This is liquid form, this is solid form. So we will do the epithelial tissue first. epithelial tissue are classified. If you want, you can, the description is on page 147 at the bottom and then 148. The words are all highlighted, okay? So let's first look at the simple epithelium. Simple means one layer of cells. Stratified means multiple layer of cells. So based on whether the cells are arranged in one layer or multiple layers, the epithelial tissue, they give names or classify based on two characteristics. One is number of layers of cells, another one is cell shape. The number of layers, whether you have one row or multiple rows, okay? So if you have only one row, they call it simple epithelium. If you have multiple rows, more than two, more than one, even if you have two rows, they call it stratified epithelium. Yeah. So based on the cell shape, it can be squamous. These cells look kind of flat, something like this, or cuboidal, where it looks like cube, or columnar, where the cells are tall like columns and they have some hairs which they call cilia, and this is your nucleus. There is some cell that looks like a wine glass, they call it goblet cell. The cilia help with movement, and the goblet cell secretes mucus. This is very typical for columnar epithelium. And then there is one we call pseudo stratified columnar. Pseudo means false. So pseudo stratified means it's not actually stratified, not multi-layers, but the way the cells are arranged, some cells are short, some are tall. But the nucleus, the position makes you think it's multi-layered suddenly when you look at it. So they call it pseudo-stratified columnar epithelium. This you have only for the columnar, so it's a variation of this. So this is like giving first name, last name. We have to consider the number of layers and the shape. Then you have either simple squamous, simple cuboidal, simple columnar, or pseudo-stratified columnar. Or you can have stratified squamous, stratified cuboidal, you don't find stratified columnar. Transitional epithelium is not really a kind of epithelium, but sometimes, let's say if you have squamous cells, when they are stretched, they look like this instead of the normal shape. So like if you have some cells here, when you do like this, if you take a section, the top will look normal, bottom will be stretched out. So it's in transition.
Okay.
The same way urinary bladder, when it fills up, it's going to stretch. So there's no technically transitional epithelium. What they have, the picture shows you a transitional phase. So I want you to look up figure 5.3, 5.4, 5.5, and 6. And all those colored figures, and try to see. First, understand figure 5.3 where they show the shapes like I have on the board. And try to see in page 149, 5.2, you can see the squamous and cuboidal, simple squamous, simple cuboidal. 150, table 5.2, you can see the simple columnar and pseudo-stratified columnar. What do you see unique there? The cilia, think of the hair, and the goblet cell, think of the wine. It's wine colored actually. Okay, the cilia and goblet cell. They are unique for only the columnar type epithelium, either simple columnar or pseudo-stratified columnar. The next set of figures just shows you their keratinized versus non-keratinized. So when they are keratinized, they look more darker, and also the shape is a little different compared to the non-keratinized. You can see the nucleus in the non-keratinized, but you don't see it very well in the keratinized, right? You don't see that dot within each cell, right? So it's full of keratin. Keratin is a waterproofing protein you find in your skin cells or hair. In the last page, table 5.3, page 152, you can see the stratified cuboidal epithelium surrounding those little space there. Like if you see a little space, you can see 2 layers of the cuboidal epithelium. Okay, then connective tissue. Connective tissue basically connects your epithelial tissue to whatever is inside your body, or it connects different parts like the bones. They're connecting the bones, or they come in between the bones, or it's basically like a filler material, like a junk or jello like substance. Okay, so you can see various functions for the connective tissue. Binding organ support, physical protection, immune protection, movement, storage, heat production, transport. Those are all functions of connective tissue. Then the cells that make the connective tissue, if you look under fibrous connective tissue, we call them fibroblasts. And there are some more cells that are involved in immune protection: macrophages, leukocytes, plasma cells, mast cells. So you have some other cells found in the tissue. Then you have, I told you about the fibers. There are 3 kinds of fibers. One is collagenous, elastic, and reticular. Okay. So among these 3, the most flexible is the elastic fiber, as the name implies. And the collagen fibers are tough and flexible. The reticular is in between in consistency. They are not as thick as collagenous, and they are not as flexible as the elastic, intermediate fibers. Then you see the ground substance. Ground substance is the background, whatever material that fills the background. We also call it matrix. Then let's look at the types of fibrous connective tissue. Overall, they classify the connective tissue into 2, loose and dense. The loose and dense refers to loose arrangement or dense arrangement, how tightly packed. So you have some examples under loose connective tissue. You have the areolar and reticular. If you see, your areolar tissue will look like this: few cells, few fibers, they're randomly arranged. Reticular will be, the fibers will be more thick. You have pictures in the following pages. And then you have dense regular connective tissue and dense irregular connective tissue. So the dense connective tissue, if it is regular, the tissue will be very dense, the packaging of the fibers. They'll have the same pattern, so they call it regular, the dense regular. The dense irregular, if you see, the fibers will be much thicker than these, and they'll be randomly arranged, irregular arrangement. So let's take a look at figure 5.14 and 15, 5.4, 5.5, and 5.6. 5-4 you can see the areolar and reticular. Can you see the difference? The reticular, the fibers are kind of thicker than the areolar, and you see much fewer cells, or you can say even both of them look kind of alike, not the color, just the arrangement. But the fibers are a little thicker. But you look at the next page, the dense connective tissue, the fibers are even more thick, right? And in one case you see the wavy pattern, it goes all the way down, the same pattern. So you have a regular arrangement, they call it dense regular. The fibers are arranged in a regular pattern, and you can compare with the dense irregular. The fibers are thick, but they are shorter and they are irregular in arrangement. Then you have adipose tissue. Adipose tissue is your fat tissue that's made up of just the cells, like in this case. You don't see the background at all, or maybe very little. And the cells of the adipose tissue, they call them adipocytes. And we are going to stop there. Okay. From cartilage, we will continue when we do the bones. That will be discussed in the bone chapter also, but we will come back and look at some of the pictures. And then we will do the muscles when we do the muscles, and nerves when we do the nervous system. So for this chapter, you need to know what is the tissue, the major categories, and some of those definitions, and what are the different categories of the epithelial tissue, on what basis the epithelial tissue is classified, what does each one mean, and then for the connective tissue, how they are classified into loose and dense, and what are the features they look for. They look for the cells, the matrix, and the fibers. Based on these three, they give names, how they are arranged. Whether you have more matrix, more cells, or less fibers, more fibers, whether they are densely packed or loosely packed, regular versus irregular, that's how they get their names. Okay?
So we have to know the description, of it,
Whatever I explained to you.
Right.
Is there anything that you'd like us to bring to class on Tuesday?
You don't need